So the cholesterol, the de novo pathway, or this synthesis of cholesterol is known as a de novo pathway, which is happening in the cytosol of almost all the tissues of the body, especially in the liver, the intestine, and skin, right? And it is a reductive biosynthetic reaction, and the parent molecule is the acetyl-CoA. Around 18 number of acetyl-CoA molecules are required for the synthesis of one molecule of cholesterol. Since it's a reductive biosynthetic reactions, the reactions are almost, or the enzymes are almost utilizing NADPs as a coenzyme, and the synthesis, the de novo synthesis of cholesterol is a high energy requiring pathway. It requires large number of ATP molecules, and the pathway, the whole molecule is carrying out its synthesis with the help of 30 different enzymes, 30 different enzymes. Almost all of them are cytosolic, except for the hydroxylation reaction, which is carried out in the endoplasmic reticulum, right? And the pathway, since it's a purely cytosolic pathway, and the pathway involves 5 different stages. For simplicity, we can divide the synthetic pathway, the de novo pathway, into 5 different stages. Stage 1 is the formation of 6-carbon compound known as mevalonate. The second stage involves the formation of the formation of isopentenyl pyrophosphate, a pentene molecule, an isoprenoid side chain molecule. The third stage is the formation of a cyclic compound known as squalene, and the fourth stage is a cyclization of squalene. Is not a cyclic structure, so it is it is a precursor for the cyclization, or we know that the cholesterol molecule is made up of 4 different ring systems. So actually the cyclization is initiated from the squalene molecule. So the third stage is one of the most important steps, and it is ending up with the formation of a compound known as squalene, and the squalene then undergoes cyclization to give rise to lanosterol. That forms a stage 4. Formation of lanosterol from squalene for the forms the fourth stage, and the last stage is the interconversion of lanosterol. Once the lanosterol is formed, it undergoes a series of the interconversion reactions to give rise to the final molecule, the cholesterol, right? So the stage 5 is actually a series of steps involving the modification of the reactions. There is a large number of interchanges, molecular changes, or modification reactions, or interconversion reactions to finally give rise to the cholesterol molecule. So actually the synthesis happens in the first 4 stages. First stage is the formation of HMG-CoA, or mevalonic acid, the 6-carbon compound known as mevalonate or mevalonic acid, and this formation of the mevalonate is the rate limiting step of the pathway. Formation of mevalonate from HMG-CoA is the rate limiting step of the pathway. So the conversion of HMG-CoA, you might be familiar with the term HMG-CoA that have already studied in the ketone body synthesis, right? And there we have told that the HMG-CoA, again we know that it is synthesized from acetyl-CoA molecules. So the acetyl-CoA molecules condenses together to first give rise to acetoacetyl-CoA. Acetoacetyl-CoA again adds one more molecule of acetyl-CoA to give rise to beta hydroxy beta methyl glutaryl CoA, or it is also known as HMG CoA, by the enzyme HMG CoA synthase. But the difference is that the HMG CoA synthase are having 2 isoforms, a cytosolic fraction and the mitochondrial fraction. The cytosolic fraction is responsible for the cholesterol biosynthesis, while the mitochondrial fraction, which is present in the hepatocytes, is responsible for ketone body formation or ketone body synthesis. And again, we know that ketone body synthesis happens only in the hepatocytes. No other tissues can synthesize ketone bodies. Again, another peculiarity is that liver never utilizes ketone bodies. Even though it is a synthetic site, it never uses ketone bodies. It always, once the ketone bodies are synthesized, it will be immediately distributed to the extrahepatic tissues. No single molecule of ketone bodies are utilized by the liver. Okay, so the mitochondrial fraction is present only in the hepatocytes, while the cytosolic fraction is cytosolic fraction of HMG-CoA synthase is present in almost all the tissues of the body, and it is responsible for the cholesterol biosynthesis. So that's a basic difference between the cytosolic fraction and the mitochondrial fraction of HMG-CoA synthase. There are two forms of HMG-CoA synthase. Again, I repeat, there are two forms of HMG-CoA synthase. One is the cytosolic fraction and the other one is the mitochondrial fraction. Both again, we know that liver is the principal organ for both the cholesterol synthesis, and ketone body synthesis happens only in the liver or in the hepatocytes. So in the hepatocytes you can see both the cytosolic fraction and also the mitochondrial fraction, but the mitochondrial fraction can synthesize only the ketone body, and the cytosolic fraction can synthesize only the cholesterol biosynthesis. So the liver synthesizes both the cholesterol and the ketone bodies, but even though it's the same enzyme, but the isoforms are entirely different. The mitochondrial fraction can synthesize only the ketone bodies or ketogenesis, while the cytosolic fraction can synthesize or can carry out only the cholesterol biosynthesis. Okay, so that forms the first step, that is formation of mevalonate. Again, mevalonate is a 6-carbon compound which is synthesized from HMG-CoA. Cytosolic HMG-CoA synthase catalyzes the formation of HMG-CoA from 3 molecules of acetyl-CoA. Then what happens? This HMG-CoA is converted into the mevalonic acid or mevalonate, and this conversion of HMG-CoA to mevalonate forms the rate-limiting step, or that is catalyzed by an enzyme known as HMG-CoA reductase. So HMG-CoA reductase is the rate-limiting enzyme of the pathway, not the HMG-CoA synthase. It is the HMG-CoA reductase that determines whether this cholesterol has to be synthesized or not. Okay, so that forms a stage 1. Then stage 2 is conversion of mevalonate to isopentenyl pyrophosphate. So pentene, that is, it's now having a double bond, isopentenyl pyrophosphate, and also there is another isomeric fraction also. Then third one is the third stage is the conversion of IPP, isopentenyl pyrophosphate, to what? Squalene, right? Then next one is the fourth stage is the cyclization of squalene to give lanosterol, and the fifth stage is the interconversion reactions to finally give rise to the cholesterol molecule. Okay, so for production of 1 molecule of cholesterol, we need 18 molecules of acetyl-CoA. It's not moles. Moles is it's wrong. You should never write mole. It is molecule. Okay, actually the slide contains molecules, moles, but I cannot correct it because it's already stored, right? So I cannot do any manipulations over it. So production of 1 molecule of cholesterol. It's not 1 mole. 1 mole means Avogadro number of molecules. This is wrong. It is 1 molecule of cholesterol, right? So it requires 18 molecules of acetyl-CoA, 36 molecules of ATP, and 16 molecules of NADPH. So that much is the investment for 1 molecule of cholesterol. And the reactions, and the slide is written as 4 stages, but in the notes, in my notes, I've divided the stages into 5 different areas. That is conversion of acetyl-CoA to mevalonate. Then mevalonate is converted to isoprenoid units, that is isopentenyl pyrophosphate, and it is also undergoing isomerism to give rise to another derivative, an isomeric form of IPP, isopentenyl pyrophosphate, to give rise to dimethyl allyl pyrophosphate, DPP and IPP. That forms the second stage. Then third stage is the formation of squalene. Fourth stage is the squalene into lanosterol, and the fifth stage is lanosterol into cholesterol. Okay. Conversion of lanosterol to cholesterol or squalene to cholesterol. So HMG-CoA is the first, again the first stage, that is formation of mevalonate, can be divided into 2 different steps. That is formation of HMG-CoA followed by HMG-CoA conversion to give rise to mevalonate. So this conversion is conversion of HMG-CoA to mevalonate is the rate limiting step of the pathway. Mevalonate is a 6-carbon compound. This 6-carbon compound then undergoes decarboxylation to give rise to 5-carbon compound. So the 5-carbon compound first formed is the isopentenyl pyrophosphate. Then 1 more molecule of IPP, isopentenyl pyrophosphate, undergoes isomerism to give rise to dimethyl allyl pyrophosphate, that is DPP. So IPP and DPP. This 1 molecule of IPP and 1 molecule of DPP, both are 5-carbon compounds. This 5 plus 5 give rise to 10. So 10 carbon compound. This 10 carbon compound again, then simple addition is happening, which is known as farnesyl pyrophosphate. Farnesyl pyrophosphate adds one more molecule of IPP to give rise to a 15 carbon compound known as geranyl pyrophosphate. So that is 15 carbon compound. This 15 carbon geranyl pyrophosphate is converted to give rise to or farnesyl the IPP and DPP just give rise to geranyl pyrophosphate. Geranyl pyrophosphate is converted into FPP, farnesyl pyrophosphate, which is a 15 carbon compound. Farnesyl pyrophosphate is a 15 carbon compound, while geranyl pyrophosphate is a 10 carbon compound. So from where does the 10 carbon compound is formed? 5 plus 5 gives 10. That is, which is a 5 carbon molecule, one molecule of IPP, isopentenyl pyrophosphate, and another molecule of IPP, but there IPP is converted into its isomeric form known as DPP, dimethyl allyl pyrophosphate. So IPP, in the glycolysis we have studied one molecule of DHAP and glyceraldehyde give rise to glycerol 3 phosphate. So similarly, in the case of the cholesterol biosynthesis, one molecule of IPP, 5 carbon compound isopentenyl pyrophosphate, will be condensed with one molecule of its isomeric form, that is dimethyl allyl pyrophosphate, DPP. So IPP and DPP adds together. So 5 plus 5 gives 10. That 10 carbon compound we call it as geranyl pyrophosphate. This geranyl pyrophosphate add one more molecule of what one molecule of IPP or gives to give rise to what the farnesyl pyrophosphate. IPP give rise to farnesyl pyrophosphate. So one molecule of geranyl pyrophosphate then adds one more molecule of IPP. So geranyl pyrophosphate is 10 carbon compound. This 10 carbon compound adds one more molecule of 5 carbon. So what is that? It is, so what happens? 10 plus 5 gives 15. So 10 carbon geranyl pyrophosphate adds one more molecule of the 5 carbon compound IPP, isopentenyl pyrophosphate, to give rise to 15 carbon farnesyl pyrophosphate, that is FPP. So now we are having one molecule of 15 carbon compound FPP. Now two molecules of FPP, that is 15 into 2 or 15 plus 15 gives 30 carbon squalene. So FPP then converts to give rise to squalene by adding one more molecule of FPP to it. So that is the synthesis of squalene. Then the squalene is then converted to lanosterol. Lanosterol is then undergoing several steps of interconversion reactions to finally give rise to cholesterol. So these are the 5 stages of cholesterol biosynthesis, the de novo pathway of cholesterol biosynthesis. See, acetyl-CoA, 3 molecules of acetyl-CoA in stepwise manner to give rise to HMG-CoA. That is, 2 molecules of acetyl-CoA give rise to acetoacetyl-CoA. Acetoacetyl-CoA adds one more molecule of acetyl-CoA to give rise to beta-hydroxy, beta-methyl glutyryl-CoA, that is HMG-CoA. This HMG by HMG-CoA synthase enzyme. This HMG-CoA synthase HMG-CoA thus formed undergoes a reduction reaction by HMG-CoA reductase enzyme. Right? It's an NADPH requiring enzyme, or it is a reductive biosynthetic reaction. Thereby reduction happens to HMG-CoA to give rise to mevalonate. And this forms the rate limiting step of the pathway. This mevalonate then undergoes conversion or decarboxylation to give isopentenyl pyrophosphate. There is 2 molecules of isopentenyl pyrophosphate will be formed. This is mevalonate is a 6 carbon compound. 6 carbon compound undergoes decarboxylation to give 5 carbon compound. Then one more mevalonate undergoes the same reduction, same decarboxylation reaction to give rise to one more molecule of isopentenyl pyrophosphate, IPP. And the second IPP will be isomerizing to give rise to DPP, that is dimethyl yellow pyrophosphate. So one DPP and IPP joins together to give geranyl pyrophosphate. Geranyl pyrophosphate adds one more molecule of IPP to give rise to farnesyl pyrophosphate, FPP. 2 molecules of FPP joins together to give squalene. Right? That forms the 4th step, squalene to lanosterol, lanosterol to cholesterol. Yeah. So this is how the reaction is initiated. The acetyl-CoA, this reaction we know. thiolase enzyme, acetyl-CoA, HMG-CoA synthase give rise to HMG-CoA. Up to this, the same reaction as in the ketone biosynthesis, but it is cytosolic. That's the only difference. This is a cytosolic reaction, while the other is a mitochondrial reaction. After this, the reduction happens to give rise to mevalonate.